forward here. So today, we're going to continue that conversation. And so we're looking at this idea of energy, right? We know that the energy currency of the cell is ATP, right? So in all living cells, energy is packed into these ATP molecules, right? We looked already at the mitochondria as the intracellular location for where this energy is generated and stored, all right? And the energy stored in ATP is available for immediate use, or it's stored temporarily until the cell needs it, all right? We in our bodies can store energy in the form of our fats and starches, right? So we know that we're using food as a source of energy, and cellular respiration, which takes place in the mitochondria, is how we get the energy that's trapped in those food molecules to a usable form, okay? ATP. So ATP is a nucleotide consisting of three main parts, right? So this one is adenosine triphosphate, all right? So we talked about the structure of a nucleotide already, where it has a sugar group, a phosphate group, and a base, all right? The base adenine, right, is going to be present in ATP, okay? And this ATP has three phosphates attached to it, right? That's that triphosphate. So ATP stands for adenosine triphosphate. Jot that down in your notes. All right. And so this is, in fact, the energy currency of our cells. And so what happens is energy in ATP is donated through the transfer of that phosphate group. And that process is known as phosphorylation, right? So this is a chemical reaction in which a phosphate group is transferred to another compound, right? So in this cycle, so we've got some energy conversion cycles taking place in the cell. Phosphorylation occurs when a phosphate molecule is transferred to ADP, right? So ADP exists and it becomes ATP during this cycle in which another phosphate group is transferred to this ADP, an adenosine diphosphate, and it becomes adenosine triphosphate. And this now becomes energy that is available to do work. All right, so we require ATP for work. What did I say to write down? Oh, adenosine triphosphate. ATP. Okay. All right. And so then we're going to shift to this concept of enzymes. So we need energy to do work, but then there's another molecule that can assist with these chemical reactions, right? And these are enzymes. Enzymes are proteins, right? A method for cells to regulate the rate of a chemical reaction can be affected by the presence of enzymes, right? So yes, we've got this energy requirement, but we can also alter or speed up the rate of the chemical reaction by the presence of enzymes, okay? Write this term down, enzymes. Enzymes are biological catalysts that function by increasing the speed or rate of a chemical reaction, right? And so what we're saying here is that cells require a constant release of energy. We know that, right? In order for any of the cellular activity, the chemical processes that are constantly going on in our cells, in order for that to occur, ATP must be available, okay? And so we must be able to regulate the release of energy to meet the energy requirements for metabolism, okay? And so enzymes are a key point of control for responding to the energy needs of the cell, right? So when we need things to happen very quickly, right? We need these chemical reactions to happen very quickly. Then we rely on biological catalysts to facilitate that process. So let's look at how enzymes work, all right? Enzymes are biological catalysts that increase the rate of a chemical reaction, okay? So the energy that we need to carry out this reaction is already there, but if we want to make this process happen even faster, we rely on enzymes. So there is an idea called activation energy, okay? And so this activation energy is the energy that's required for this reaction to take place. And so it's sort of the energy barrier or threshold that must be met. All reactions are going to have this energy barrier or this energy threshold that is required, all right, in order for this chemical reaction to take place. That is called the activation energy. Write that down. You need to understand, what is the activation energy? Activation energy is the amount of energy that's required at baseline for this chemical reaction to take place, okay? This is independent of the enzyme itself, okay? Every chemical reaction has a required amount of energy that's necessary for the reaction to take place. That is the activation energy. And so what enzymes do is they come in and they actually lower or reduce the amount of activation energy that's required for this process to take place. And it essentially speeds up the chemical reaction by reducing or lowering the amount of activation energy that's required. So in your notes, what I want you to understand is that for a chemical reaction to take place, metabolism, right? We're just talking about any type of chemical reaction that's taking place in the cell. In order for that chemical reaction to take place, it requires energy. It has a certain amount of energy that must be present. That is called activation energy, okay? Enzymes come into the equation as a mechanism for lowering the amount of energy that's required for this chemical reaction to take place. So we say that enzymes speed up chemical reactions by lowering the amount of activation energy that's necessary to initiate that chemical reaction. That is a very fundamental concept that I want you to understand. What questions do you all have about this? So, in your own words, you should be able to tell me what is the relationship between enzymes and chemical reactions, okay? Why are enzymes important in the cell? Why do we need enzymes? Why are these enzymes that are referred to as biological catalysts, right? A catalyst is something that initiates, right, or starts something. Why are these biological catalysts important? Okay. You must understand what an enzyme is and its job as a biological catalyst. This catalyst is speeding up this reaction. Okay, so let's look at this schematic here. Let's stay in this left panel right here. All right, so these are examples of a chemical reaction taking place, right? We've already talked about the structure of a chemical reaction and how we have our chemical equations that represents what's going on in a chemical reaction. So we have our reactants on one side of the equation, and then we have our products on the other side. And there is a conversion that takes place, right? We have a change in our substances. We start with certain reactants and then we end up with different substance is being generated. So in a chemical reaction that is uncatalyzed, right, that means there's no enzyme present. All right, so on the x-axis here, we have our uncatalyzed reaction. On the y-axis here, we have the amount of energy that is being used. So we have our reactants that will now become products, right? There's a normal amount of activation energy required to catalyze this reaction, where we're going from reactant to a product. And we see how much energy is going to be necessary for this reaction to take place in this uncatalyzed reaction. No enzyme present. Now, over here, we have a catalyzed reaction, the same chemical reaction where we have reactants that will become new products, all right? However, in an enzyme catalyzed reaction, we have a reduction in the amount of activation energy that was necessary, okay? So the activation energy was lowered by the presence of this enzyme, okay? So enzymes function by lowering the amount of activation energy that's required to catalyze a reaction. It is important that you understand this concept. What questions do we have about these schematics? If I showed you something like this, you should be able to interpret what's happening. On the left, we have an uncatalyzed reaction. And on the right, we have a catalyzed reaction. And if we look at the total amount of energy that was necessary for this reaction to take place, we see that over here in our enzyme catalyzed reaction, the amount of energy was significantly lower than that in our uncatalyzed reaction. So this reaction over here on the right is going to take place at a much faster speed, or this initiation of this reaction is going to take place a lot faster. Okay, it's going to speed up. Okay, due to the presence of this enzyme. What questions do we have about this? Okay. So how do enzymes function? Enzymes require a substrate, right? And we create something that we refer to as an enzyme substrate complex. Okay. So enzymes influence reactions by forming a complex with a substrate. Okay. The substrate is the substance on which it is acting, right? So in order for this enzyme to have an effect in this chemical reaction, it's got to bind to something, and it's binding to the substrate. Write that down. Enzymes bind to a substrate, and this is the substance for which it is going to act upon. Okay. And the place where this enzyme interacts with the substrate, it's called the active site. So think of it kind of like a lock and a key, all right, or puzzle pieces fitting together. The active site is the region where the enzyme binds to the substrate. So it is a very specific site, very specific shape or conformation, right? So remember, our enzymes are proteins, and we talked about the different shapes and conformations of proteins. And so this enzyme is going to be of a specific shape, and it's going to have to fit precisely into another specific groove on its substrate. Okay, that's called the active site. All right, so we're getting some new terms here. Enzyme substrate complex is formed as the enzyme is acting as a catalyst. Okay, the enzyme has to bind to the substrate. There's a very specific site on the substrate called the active site where this enzyme is going to bind. What questions do we have about this? Any questions? Okay.
No, not right now.
Okay. And so here's our visual, right? So we have our enzyme, right, which has a specific shape or conformation, and then it's got some substrates for which it's going to act upon. All right. So in order for us to get this enzyme substrate complex happening, right, the substrates bind to, or the enzyme binds to specific sites on the substrate known as the active site. Once this enzyme substrate complex is formed, the chemical reaction can commence, and we're going to go from reactants to products. Okay, so we need to have an enzyme interacting with the substrate. We get the enzyme substrate complex, right? And now we've got new products being formed, right? And this enzyme is going to be released to go off and do the same thing again. All right. And the point you're noting is that the enzyme binds on the active site to form this enzyme substrate complex. So in order for us to go from reactant to product, we've got to form this enzyme substrate complex first, sort of as an intermediate process. Okay. So enzyme specificity, write this down. Enzymes are very specific. Okay, so the shape of the active site closely resembles the shape of the substrate. So remember, I mentioned kind of like a puzzle piece fitting together, right? Most enzymes are going to be highly specific. Okay, they're only going to catalyze a very few types of reaction, or one particular reaction. So each reaction might have its own enzyme that it requires. Okay, so enzymes are very specific. Those active sites must be the right shape, okay, in order for this enzyme to bind to get this enzyme substrate complex formed. Okay, so the point you're jotting down is that enzymes are very specific. Each reaction has its own specific enzymes that's going to catalyze this specific reaction. So no enzyme is going to be just universally able to catalyze any type of a reaction, right? They are very specific to the types of reactions that they can catalyze by way of that active site on the substrate. Okay, so the enzyme shape must complement the shapes in that substrate, those sites in the substrate, in order to bind and become active. Okay, so we're still talking about chemical reactions here, metabolism. So there are some other factors that play into enzymatic activity, and those are cofactors. Okay, enzyme cofactors also influence our reactions. Okay, so some enzymes only contain the protein, which is the apoenzyme, right? So the apoenzymes is just basically an enzyme without a cofactor attached. Okay, some other enzymes have additional components that we'll call a cofactor, right? In this case, if a cofactor is present, neither the apoenzyme or the cofactor has catalytic activity alone. Only when they are combined does the enzyme function. Okay, so what I'm saying is that some enzymes function completely independently as an enzyme, as that protein. An enzyme is a type of protein. Some of them are active as an independent protein. It has the enzymatic activity. We call that an apoenzyme. This is just an enzyme. It's just a protein. However, some enzymes require additional chemical components, kind of like a partner. And we call those cofactors. If this said enzyme requires a cofactor in order to be active, it's not going to have any enzymatic activity without that cofactor. All right. So these are two new terms here. Apoenzyme is just the enzyme that is a protein. It's just the protein itself. All right. The cofactor is an additional chemical component or substance that's required for this enzyme to be happy and to have its catalytic potential. Okay. If the enzyme requires a cofactor, it will not be catalytically active without that cofactor. Okay. And so then there are some additional conditions that must be met in order for enzymes to have their optimal activity. Okay. And so we know that enzymes work best under certain defined conditions, and we call these optimal conditions. So we're talking about the overall environment here, right? So enzymes work best under optimal conditions. And optimal conditions are defined when the rate of the reaction is at its fastest. Right? So remember, we said the job of the enzyme is to speed up the rate of the chemical reaction. Right? It's going to do that and work best under optimal conditions. And these predetermined conditions include the temperature, the pH of the solution or reaction, the ion concentration, a number of different factors that are preset and must be met in order for these enzymes to function at their best. And again, we're talking here in a system, in a biological system where these chemical reactions are taking place in the cell. This environment has to be, these optimal conditions must be met if we're going to get these enzymes to function top notch. All right. So enzymes work best under certain predefined conditions. These conditions can be factors of the temperature, pH, concentration, a number of different factors that we can assess or can change within the cell, but that must be met in order for these enzymes to function optimally. All right. I'm going to stop here today. I know that's a lot of information. What questions do you all have?